Introduce our next new fellow, Professor David Gardner. David is a world-leading embryologist. His research has laid the foundation for major clinical developments in human IVF. Please make him welcome. David.
Thank you to the Academy. This is a tremendous honor. This is the fertilized human egg. It's the largest cell in the human female body. It's 110 microns across, so that's still smaller than the full stop on a printed page. And this cell has become my obsession for the last 30 years. An obsession that took me from England to America, from America to Australia, from Australia back to America, and 10 years ago, I'm very grateful to be back in Australia. It's an exquisite cell, and as it develops and differentiates into what we call the blastocyst, it undergoes quite remarkable metabolic transformation. So much so that the fertilized egg, metabolically, is distinct as any cell type can be from this wonderful structure which we call the blastocyst. And it's through the analysis of metabolic function that we've been able to understand how we can grow these cells, but also we've learned there's a lot more to metabolism and how metabolism is used by these cells to interact with their environment. Now, when I talk about metabolism, most people think of this, and we go to shudder down our spine. There's lots of reactions going on, and A goes to B, and it's fluxes of carbons and nitrogens, and the occasional molecule of ATP comes off. I don't think it's really that helpful as a diagram. I think this works much better. Because you can see it conveys things to you. You know this is about a flux. It's about, in this case, a flux of trains carrying people, but rather than people, metabolic pathways carry carbon and nitrogen. And at different times of the day, the different lines are more active than others. And in this age of social media, the very things that have been passaged around, the passengers, are also sending out information about their journey. And so now we're getting feedback from the passengers to the control about what's happening. And this is fundamentally different, and this is what's happened to our understanding of metabolism. The passengers, or in this case, the carbon molecules that are getting passed through metabolic pathways, are actually acting as signaling molecules too, and are taking more control of the destiny of the cell. So this is the first week of life. We've all been through this. This is the wonderful cell that is the egg. It's an amazing cell because it can reside in the ovary for up to 40 years and still give rise to a viable pregnancy. So metabolically, it's adapted to this role. What it's done, it says, right, I can't use glucose, I can't use much oxygen, because you don't want oxidative metabolism, because that will create reactive oxygen species, and I'm trying to protect these delicate chromosomes on this spindle in my egg. So it uses low levels of oxidation of carboxylic acids. But by the time it develops and differentiates into this beautiful blastocyst on day 6, day 7 down here, it has become the most active tissue in the body. And it consumes lots of glucose, and it has a very high oxygen consumption. But with a twist, it doesn't oxidize all its glucose. It produces a lot of lactate. And this is a phenomenon known as aerobic glycolysis, not the anaerobic glycolysis you got running to this lecture. It's the aerobic glycolysis, the production of lactate in the presence of oxygen. And as a graduate student, I found that observation really perplexing and wanted to know what other cell types exhibited this behavior. And the one that popped up was cancers. Cancers and blastocysts share a very similar pattern of metabolism. In fact, Otto Warburg in the 1920s discovered the fact that cancers have aerobic glycolysis, so it's also known as the Warburg effect. So very early on, I thought, well, embryos are like cancers. I was wrong, and I'll come back to that. So the significance of metabolism is we all need it to survive, but with regards to an embryo, you're changing from a quiet metabolism to a very active metabolism. And what we found out is that if the embryo loses this ability to do the transfer of metabolic pathways, we lost development, and that meant we lost pregnancies. So by understanding the significance of this switch, we were better able to grow embryos. So only 2 schematics on metabolism. But what I want to show you is that glycolysis is pretty much shut down, and pyruvate is the primary nutrient to the cleavage stage embryo. In contrast, by the blastocyst, glucose flow is very high, half of which, though, goes to lactate. And I want to bring to your attention only this one reaction. Pyruvate goes to lactate with the production of NAD. I'm going to come back to that one simple compound, NAD, because it's a game changer. So pyruvate produces lactate and NAD. The other side of this diagram is over here. It's the malate aspartate shuttle. For years, we'd worked out that amino acids regulated carbohydrate metabolism, but we didn't know how. Turns out we identified that this pathway was active both in embryos and embryonic stem cells. And it is absolutely fundamental to their physiology and survival. The other thing we were able to do was work with physicians who took samples of oviduct and uterine fluid for us so we could determine what exactly did the human embryo see in the mother. And as you can see, the early embryo doesn't see very much glucose. It's the later stage embryo that sees the glucose. So what we can derive from this is that even before implantation, mom knows what's best for her embryos and provides these nutrient gradients. The significance of this was, up to the mid-90s, we could not grow the human embryo past the 8-cell stage. And so in human IVF, we were forced to put these cleavage stage embryos back prematurely into the uterus, which is a different environment. The result of that, if you did that in an animal model, would be very low pregnancy rates. And in fact, human IVF was marred by very low pregnancy rates for a long, long time. To compensate for that, more than one embryo was routinely put back. 2, 3, even 4 were put back. So we had this multiple gestations. So infertility actually provided us with the highest number of multiple gestations. By examining the metabolic status of these cells, by understanding their changing physiology, we were able for the first time to routinely grow the human embryo to the blastocyst stage. This meant that we could transfer single embryos because their implantation rate was much, much higher. So now, rather than having multiple gestations, we just have singleton pregnancies by the transfer of this. And this technology has resulted in the birth of more than 1 million children to couples who would have been childless without this blastocyst transfer technology. And I'm pleased to say this is the standard of care now in Australia and New Zealand. The other thing we're able to do is, if you look here, this is the inner cell mass. This is where the baby comes from. In the mid-90s, I was in America presenting this story, and a group of stem cell biologists was in the audience. And the year before, they'd isolated primate stem cells, embryonic stem cells, but couldn't grow the human embryo to do embryonic stem cells. So we provided them the technology to grow human embryos, and within a year, the first human embryonic stem cells were identified. We can use these nutrient uptakes to measure the activity of embryos before we transfer them, and it appears that the more viable the embryo, the more nutrients they consume. To do this, we need to use really complicated analyses, but we worked with a group at MIT to create a microfluidic chip. And these chips over here, this is 3 nanoliters of culture medium being mixed with reagent on a little microfluidic chip, and we can automate how we quantitate now nutrient uptakes. So I think this is going to be the future, using microfluidic devices to assess the health of embryos. Now, it's often overlooked that oxygen is a very important role. All of us in this room are breathing around 20% oxygen, but the only part of our bodies that sees that are the lining of our lungs, our skin, our eyes. At the capillary bed, it's around 10% oxygen. In fact, the oviduct and uterine levels are about 5%. And yet, most of the world's IVF, in fact, most of any tissue culture, whether it's cancer biology, MS, uses 20% oxygen. And actually, oxygen is a very powerful regulator of metabolic function. This shows the utilization of amino acids at 5 and 20% oxygen, and what happens is oxygen completely changes how cells metabolize. They change glucose. They suppress the very marker of viability. So it's very important to put that into context. So this is conventional signaling, how we see a linear trend towards what controls metabolic function. Well, actually, we're now looking at this paradigm shift where external metabolites and cofactors impact metabolic function, which directly affects epigenetic regulation. And this is the so-called metaboloepigenome. And here we have this beautiful little molecule of NAD again, affecting what we call the SERTs, which can regulate acetylation of histones and therefore gene expression. So this is a really exciting area for the future. I just want to spend a couple of minutes on this aerobic glycolysis of the blastocyst, because it's intrigued me for so long, and I thought I got it right. If you've got aerobic glycolysis, it means you're able to put a lot of glucose through the pentose phosphate pathway. You're able to generate ribose moieties for RNA and DNA. And I was quite happy with that. And in many ways, I thought blastocysts were like cancers. Implantation is quite analogous to invasion. So in this play, there are 3 acts: breakdown of the extracellular matrix, induction of angiogenesis, and modulation of the immune system. And in today's play, the role of the blastocyst will be played by myself, and the role of the endometrium will be played by all of you. Here I am. I'm consuming a lot of glucose. I have the carriers to both take it up and export lactate. I'm consuming a lot of glucose. And as a result, I'm creating basically an acid cloud around me. Now, we're in this very big room, but cells exist in microenvironments, so please imagine now we're actually in an elevator. We're in very close proximity. I'm producing a lot of lactate, and ladies and gentlemen, whether you like it or not, I'm going to make you eat my lactate. You have no choice. But the result of which is fundamentally, I'm going to change your physiology, okay? If you want to break down a tissue, low pH upregulates the activity of metalloproteinases and other enzymes which break down tissues. It also increases the synthesis of hyaluronic acid. Hyaluronic acid is unique in that it can absorb water to thousands and thousands of times more than its own mass, so it spreads out. So I have the effect of literally being between you and pushing you all apart, which is great from trying to break you down. And finally, we get the upregulation of these proteases, but we also downregulate your ability to turn my proteases off. Angiogenesis, we need to create a blood supply. If you cut yourself, you break the microvasculature. The first thing that happens is that those tissues become anaerobic and produce lactate. Lactate is the key signal to induce blood vessel formation. So by putting out lactate, I'm going to take endothelial cells, they're going to come to me, but also they're going to make tubules. I have this other capacity. Remember that NAD from the pyruvate. Now, I'm putting lactate down your throat, but I'm making you consume your own NAD. You don't have it. NAD is very important because it's also used in ribosylation of other proteins. Vascular endothelial growth factor, which is produced, is ribosylated by your cell, and it's inactivated. By turning that capacity off in your cell, I'm upregulating bioactivity. The site of implantation is propensity of macrophages. I can convert an M1 hostile to an M2 phenotype simply by putting lactate in your environment. So we've changed that entirely. So rather than thinking that the embryo is like a cancer, in fact, cancers are like an embryo. Okay, so now we've actually made synthetic blastocysts to take that out. So it's my pleasure to acknowledge the ARC, Stem Cells Australia, who've supported a lot of our work. Vitrolife has been a commercial partner with us to translate our stuff. This is my current team. I would also like to thank the many, many, many graduate students and fellows from around the world over the last 30 years, and in particular my mentor, Henry Lees, who prepared me so well for a 30-year journey in obsession. Thank you so much.
Thank you, David. Are there any questions? We have time for a couple of questions. Yes, Philip?
Philip Crucial, Sydney. What's the lactate receptor?
The lactate receptor, the cells all have the monocarboxylic transporter. And so by mass action, we can drive it back into the cell. So it's not exactly a receptor, it's the fact that we all have those transporters. And typically, we can drive the lactate out, but mass action will determine that we can push it in to the other cells.
There is also a lactate receptor in glomus cell.
Okay.
I reckon you can find it.
I look forward to looking for it. Thank you.
There are no more questions. I'd like to ask you to join me in thanking David for a very nice talk.
Thank you.